Welcome back friends, welcome to another tutorial from Just Biology. In this series of lecture, we'll be discussing various topics in rDNA technology. Probably this is the first lecture on the rDNA technology. We want to talk about a little bit of rDNA technology. This might be an introduction of the rDNA technology. As far as the rDNA technology is concerned, what are the things actually we should know? That is what we are going to discuss in this lecture, and upcoming lecture we will discuss some other topics or various things in rDNA technology. Let's begin with, as it is rDNA technology, we need to know the recombination is a process by mixing or the blending of two different DNA. The two different DNA will have two different function as well. That's why we call it as rDNA, so the recombinant DNA technology. Whenever we start with rDNA technology, we will also refer some kind of experiment. Then what is that experiment, or what is what it means? The first thing, the recombination will happen actually by by natural also it is happening. How the in laboratory condition, how we can make the recombination? So that is what we are going to discuss. The process of cloning. So particularly we are going to discuss cloning only in this lecture. So just introduction to the cloning. The cloning is a process by which we will make many copies from a single gene. That is, let's say here we can take X. Just imagine this is a gene. I want to prepare X gene into many copies or so many copies. What I need to do? I need to replicate or I need to duplicate them. By natural way, actually they did naturally. But how artificially I can make this happen? That process is called as cloning. So in this cloning, we'll use two things. One is host and the second one is vector. I'll tell you what it means actually. The vector and host. The vector means the gene or the fragments of the DNA which carries into the cells, or the place of interest, the where we want to transfer. So those things will be referred as vector. Then probably what is host? The cell which we will transfer the DNA, that cells is known as host cells. The cell which receive the vector is known as host cell. The host is probably a bacteria because they have a plenty of properties like generation gap. The first thing is generation gap. Gen gap. So what do you mean by generation gap? Generation gap is a time period or the time taken for multiplication or the duplication. That is referred as generation gap. The host of the bacteria, the bacterial host will have a less generation gap. If you take fungi, that would take minimum 3 days for multiplication or their growth. But in case of bacteria, they have only 20 minutes. They have only 20 minutes to multiply. Example for this is E. coli. If you take E. coli, and you can go for checking also. So which will give the cells within 20 minutes, that can duplicate within 20 minutes of incubation time. So this is actually we should know the vector, host, then the duplication. How this vector will take up the gene of interest or the fragments which we want to multiply. How the cloning is organized, so that is what we will discuss now. The organization of this cloning So that is referred as several steps. Actually, normally the 5 steps they have. The first, what we need to do, we need to get the DNA which we want to duplicate, or the fragments. Then after getting the DNA, we should select the vector. So that is important here. What kind of vector? You can go for the vector which I mentioned earlier. The first step, just remember, we need to collect the DNA. The second step of cloning is we need to select the vector. Once you got the DNA as well as the vector, then what you need to do? You need to restrict both of them with the same enzyme. So here you should know the enzymes here. In case of cloning, they plays a major role here. What kind of enzyme we usually use here is nuclease. Nuclease is an enzyme which degrade or which restrict the DNA molecule or the RNA, because they belongs to the nucleic acid. The DNA as well as the RNA belongs to the nucleic acid. So they cut the nuclease for both DNA as well as the RNA. In nuclease, they have various types of nucleases are present, in which endonuclease are most important. Endonuclease. Endonuclease. The endonuclease will cut the DNA based on the specific size which they have. So that's why the endonuclease is more important here. Then once you got the DNA fragment as well as you restricted the vector, what you need to do? Then you need to join both the fragments as well as the vector. Both as vector as well as the fragments which you digested. For that also you will use this enzyme is called as ligase. That enzyme will join the DNA. Ligase, the enzyme, the ligase is an enzyme which join the DNA, because they tend to induce the formation of phosphodiester bond between two nucleotide. So this is actually an enzyme complex process, because once the cutting as well as the joining is mediated by enzyme. Okay, next we'll move on to the final step, or the last step. Once you got the DNA and once you join the DNA, then what you need to do? You need to transfer that molecule. Actually, let's say, so this is the fragments which you obtained from a genome, any genome. Then this is actually, so this is a vector. You digested with the same enzyme for both gene as well as the vector. The first step, just I mentioned earlier, will join together. This vector will join together. So this is called rDNA. This is what it means, rDNA, recombinant DNA, because you blend two different kinds of gene information or gene. So this rDNA molecule will be transferred to the host. So that is what the last step, not last, second last step. Host, then the host will multiply. The host will multiply, or they make many copies of the same set of information. Then after the making of all this stuff, we need to collect. We need to collect all this vector from the host cell. Once you got the DNA or the duplicated DNA or amplified DNA, then you need to go for checking whether they are transformed or not transformed. For that, we have many procedure, many technique for the identification of recombinant clones. So that will discuss in future lecture. Then now let's look at the screening. The screening is a process, just now I said. So we need to check the recombinant clones. If you checked the recombinant clones and you just identified the cells which has the DNA molecule, you can select those cells alone and you can preserve that cells. Then if you are going to make any library like you want to collect any information of various organisms DNA or the genome, you can go for library preparation. For library preparation, you can preserve these cells for a future study. Then you will keep it in some kind of some kind of chemical. With the chemical you can preserve. So this is actually a basics of our DNA technology. Our DNA technology means we want to make amplified copies or many copies of like a particular gene. So this is called cloning, and this is the basic technique of our DNA technology. Hope you understood, guys, the concept of cloning. If you like the video, please hit the like button and don't forget to subscribe my channel and click the bell icon for latest update. Keep watching, stay tuned, guys. Thank you for watching, guys.